Hi everyone, this is Vivid Botany. We are discussing NEET 2021 questions. Let us see this question. A typical angiosperm embryosac at maturity contains the number of nuclei and number of cells. This question is from sexual reproduction in flowering plants. I often call it as SR in flowering plants. This chapter is from NCERT plus 2, the 12th standard, and to have a complete understanding of the question, one should know the megasporogenesis. Megasporogenesis is nothing but formation of megaspore from the megasporangium. Megasporangium is something that we commonly call as ovule. So this I am drawing here the anatropous ovule. Anatropous ovule is 180-degree curvature it shows. This is anatropous ovule. This part is called as the micropyle, that is opening of the ovule, whereas the stalk of the ovule is called as funicle, and this is called as ovule, or we call it as megasporangium, because it produces the megaspores. The diploid tissue present in the megasporangium is called as nucellus. This diploid tissue present in ovule is called as nucellus. From the nucellus, megaspore mother cell is differentiated. Megaspore mother cell is differentiated from the nucellus. This is diploid, and this megaspore mother cell undergoes reduction division to give rise to 4 megaspores. So these 4 megaspores are haploid in nature because they are the products of RD, reduction division, or meiosis. So megaspore, as it is haploid, they are considered to be the initial cells of the gametophytes. Out of the 4 megaspores, 3 megaspores degenerate and 1 megaspore remains functional. This functional megaspore is generally present at micropylar end. It is present at micropylar end. So I have shown you that here this micropyle, here the functional megaspore is present. This megaspore undergoes karyokinesis. Karyokinesis means, remember, it's only division of the nucleus but not cytoplasm. And functional megaspore, as I told you, this is haploid. Due to the first karyokinesis, 2 daughter nuclei are formed which are haploid. Further they undergo second karyokinesis, they form 4 nuclei. Further they undergo third time another karyokinesis and they form 8 nuclei. These 8 nuclei are arranged in such a way that, remember, these are 8 nuclei, they are formed by the karyokinesis that takes place 3 times. This is 1, this is 2, this is 3. 3 times it took place mitotic division having only karyokinesis. And how many times? 3 times. Number of cell divisions are, they include here 7. Is it 1, 2, 3, 4, 5, 6, 7. 7 mitotic divisions, 3 times they took place. As a result, 8 haploid nuclei are formed. These 8 haploid nuclei are arranged in such a way that 4 are arranged in upper pole and 4 are arranged in lower pole. Now, one nucleus from the upper pole migrate towards the center and one nucleus from the lower pole migrate towards the center. As a result, what happens here? 3 nuclei are present in the upper pole and 3 nuclei are present in lower pole. 3 nuclei are present in the upper pole, 3 nuclei present in the lower pole, 2 nuclei present in center of that. Now, this 8 nuclei in total will undergo cytokinesis. That is, they form cell wall, cellularization we call it. So, here 3 cells are formed in the lower pole. Here are the 3 cells which are formed in the lower pole, and here 2 nuclei with 1 cell formed in the center, and here we find the 3 nuclei in the upper pole. Rather, these cells are very small in size when you compare with rest of the cells. So, this is the whole structure having this is called as embryosac. We call it as embryosac. So, they look like this. Embryosac we call it here. 3 cells in the lower pole, 2 nuclei with 1 cell in the center, and 3 cells in the upper pole. The 3 cells in the upper pole are called as antipodals. Antipodals. They are 3 in number. They are haploid in nature, of course, and these cells are the smallest cells of the embryosac, and they are also called as vegetative cells of embryosac because they do not participate in the so-called fertilization. And this is called as the central cell. It is called as the central cell. It is having 2 polar nuclei. It is the largest cell of the embryosac. And these 3 cells are called as egg apparatus. The egg apparatus contains 3 cells. It is the larger cell called as the egg cell, that is the real female gamete of the embryosac, and these 2 cells are called as synergids. They are called synergids. So, the whole structure is called as embryosac. It is called embryosac. It's also called as female gametophyte. It's called female gametophyte. So, this female gametophyte is in fact present inside the ovule. As I have told you here, inside the ovule we find the female gametophyte or the embryosac. So, this is here. We find here again what you call the micropyle, and this is the funicle. Inside that we find the embryo sac. So, this embryo sac or female gametophyte is having here 7 cells. You can find it here having 7 cells, like the 3 antipodals, 1 central cell with 2 nuclei, and egg apparatus having 3 cells. So, it is 7 cells, and the number of nuclei if you count, as the antipodals are 3, central cell contains 2 nuclei, 3 plus 2, 5, plus 3 nuclei present in egg apparatus. So, it is having 8 nucleated. So, very specific it is having 8 nucleated and 7 cells. The whole process, if you see in the diagram here, it is something appear like this. It is like this here. This is central cell, and these 3 are antipodals. So, this is what we call as the egg apparatus or female gametophyte. This egg apparatus is found inside the ovule. Ovule is found inside the ovary. Ovary is part of the carpel. Carpel is present in pistil. Pistil present in flower. Flower is present on sporophyte. So, sporophytic plant, flower, then carpel, in that carpel then ovary, in that ovary then the ovule, in that ovule this female gametophyte or embryosac, in that we find this, what do you call, the egg apparatus, and in this egg apparatus we find the egg cell. So, this is the whole history of this megasporogenesis. It is very particular, only seen in angiosperms. So, number of cells are 7, number of nuclei are 8. So, the first option 8 nucleated and 7 cell. Remember, it is the most important question. You should know the position of that cells also. You should know the number of cells, number of nuclei, how many RE took place here, how many mitotic divisions, how many times mitotic divisions. All this, if you know and if you observe very carefully, you can do such type of problems without any difficulty.